Actually, I'm still going to talk about signal transduction. And I am trying to present the family of signal transduction enzymes I work with. So these are called PI3 kinases, phosphoinositide 3 kinases. These are lipid kinases that are activated by different receptor types and produce a phosphorylated lipid on membranes. Usually this lipid doesn't exist, but when cells are stimulated, this lipid accumulates and starts to function as a docking site for a series of different signal transducers. And actually PI3 kinases emerged as controllers of a wide range of different biological responses. For example, PI3 kinase is activated by growth factor receptors like, for example, insulin receptor. And they are known to control a lot of different effects mediated by insulin. PI3 kinases can also be activated by G protein coupled receptors. And actually, the PI3 kinase family can be subdivided in two subgroups. The class 1A PI3 kinases are those enzymes that are activated by tyrosine kinase receptors. In the class 1B PI3 kinases are activated by G protein coupled receptors. Actually, while three different enzymes exist in the class 1A subtype, there's only one in the class 1B, the PI3 kinase gamma. And we focused our attention on this particular subtype already for quite some time. Years ago, we actually observed that mice where this PI3 kinase gamma is either inactivated or mutated to become catalytically inactive, the major phenotype is an immunological one. So basically, while most of the class 1A PI3 kinases are ubiquitously expressed, class 1B PI3 kinase is a little bit more restricted. At least it's heavily expressed by leukocytes. In the phenotype we observed in mice with carrying mutations of this gene is that leukocytes don't migrate to the site of inflammation. So basically, PI3 kinase gamma activity downstream G protein coupled receptors is needed for the recruitment of leukocytes to the site of infection or inflammation, of course. So the major phenotype was, let's say, indicating that targeting PI3 kinase gamma was going to be a promising therapeutic strategy to treat inflammatory diseases. But with time, having these mice available, we also understood that PI3 kinase gamma expression pattern is a little bit more wider than expected. So PI3 kinase gamma is not only expressed by leukocytes, but it's also weakly expressed by other cell types, for example, endothelial cells or cardiomyocytes. And as the idea was to develop anti-PI3 kinase gamma inhibitors to treat inflammatory diseases, then the concern that emerged was maybe targeting this enzyme is going to provoke serious effects on the cardiovascular system, for example. So that was actually a serious concern coming from our partners in the industry. And that is probably why we started to deeply analyze the phenotypes happening in the cardiovascular system. And these were all kind of interesting surprises. First of all, I have to say that our NOCA mice and the mice expressing this catalytically inactive PI3 kinase gamma did not show big phenotypes in basal conditions. So basically, the heart and the cardiovascular system was not that heavily affected. But we started to think that maybe we had to stress our animals to really uncover some function for this PI3 kinase isoform. And that is the reason why we decided to go for a specific model that is called transverse aortic constriction. So this model is actually a model of hypertension, of chronic hypertension. And usually this increased workload happening to the heart because we close a little bit the aorta. The heart has to pump more. And usually the heart goes into a series of remodeling steps that you initially are compensatory, but with time, this compensation gets broken, and the hearts show dilation and all the signs of heart failure. That is actually why it is believed that high blood pressure is a silent killer, because it imposes stress to our hearts, and with time we can actually develop a heart failure. So the interesting thing is that when we treated these mice in the model of aortic banding, we were able to see, at least in mice expressing a catalytically inactive PI3 kinase gamma mutant, a very striking protection. So I told you that after banding of the aorta, the hearts become more hypertrophic, but with time, the chamber of the left ventricle dilates and gets larger. So basically, the blue line is a wild-type control, and you see that the chamber is getting larger. But the orange line is what happens in our mutant mice. So basically, the chamber doesn't dilate. So these mice are protected from heart failure. The point is now, what are the cell types that are involved in this protection? Because I told you PI3 kinase gamma is important in inflammatory reactions in leukocytes, but also I started to say PI3 kinase gamma is expressed in cardiomyocytes as well. So to try to address this specific question, we studied mice that were, let's say, irradiated and where bone marrow was transplanted. So basically, we ended up with 3 groups: a control group where wild-type hearts were in the presence of wild-type leukocytes, but also we obtained all the other potential mixtures. So we had mice with mutant leukocytes and wild-type hearts, and mice with wild-type leukocytes and mutant hearts. So if we look at what happens with time, this is, let's say, a measure of contractility, so cardiac function. The green line is, of course, what happens in wild-type controls, or the mice that received, wild-type mice that received the wild-type bone marrow. So with time, these mice develop heart failure, and their contractility goes down. A little bit expectedly, mice that had mutant leukocytes that don't go to the inflammation site were protected. These mice actually didn't have leukocytes going to the heart, and this protected from development of fibrosis. And fibrosis, it's a kind of inflammatory reaction that causes stiffness of the left ventricle, and of course blocks contractility. So as you see here, mice with mutant leukocytes but wild-type hearts are slightly protected or are significantly protected. But the big surprise in a way is the pink line. The pink line is mice with wild-type leukocytes, so fibrosis goes on, everything is remodeled in an adverse way, but in the end, as you see, contractility is protected. So we started to think PI3 kinase gamma must be a function in cardiomyocytes as well, and this function must be important because these cardiomyocytes are nicely protected even in a situation where inflammation occurs, fibrosis occurs, and the contractility is usually affected. So we started to go for measurement of PI3 kinase gamma activity and compared the hearts with, let's say, a normal condition and hearts that developed heart failure, so basically after 20 weeks of aortic banding. And as you see here, the activity of the enzyme increases with heart failure. So this means that somehow PI3 kinase gamma activity is modulated in the remodeling of the heart following aortic constriction. And we think that this is indeed something crucial. We think that this is mediated by an upregulation of this enzyme in hearts. And we also think that this has a clinical relevance. Because you see here, we analyzed by immunohistochemistry PI3 kinase gamma expression in sections of human hearts of patients that presented aortic stenosis, so basically the kind of a disease, of acute disease that we wanted to model with aortic banding. And as you see here, patients with aortic stenosis show a tremendously upregulated PI3 kinase gamma expression in cardiomyocytes. So that is why probably PI3 kinase gamma activity goes up. But why should this go up? What is the biological mechanism underlying this effect seen in the heart? So the literature was some kind of helping us, and there were reports indicating that PI3 kinase gamma can be involved in not only in G protein kappa receptor signaling, but also in G protein kappa receptor desensitization and internalization. So basically the idea is that the production of PIB3 around G protein kappa, activated G protein kappa receptors allows internalization. In the internalization of G protein kappa receptors, that in the heart, the best known are of course the adrenergic receptors, the beta adrenergic receptors, is a crucial event in heart failure. It is well known, both in mice and in humans, that shedding of adrenergic receptors is a typical hallmark of heart failure. And actually, one of the effects of the few drugs that help patients with heart failure, beta blockers, in the long run, their effect is to restore adrenergic receptor densities at the plasma membrane. So as expected, here you see wild-type mice before and after developing heart failure. And as expected, you see that the number of exposed beta adrenergic receptors is significantly decreased when mice develop heart failure. The interesting thing is that if you look at mice that express the catalytically inactive PI3 kinase gamma, then you see that this downregulation of adrenergic receptors doesn't occur. So basically, we think that blocking PI3 kinase gamma activity has a specific role in reorganization of beta adrenergic signaling in the heart. And actually, we think that this has some clinical relevance as well, because we have some already significantly selective inhibitors developed by our collaborators in the industries that block PI3 kinase gamma activity. And you see here we gave these inhibitors to wild-type mice after development of heart failure. And as you see here in the green line, the amount of adrenergic receptors is increased after treatment of these mice for 15 days with the PI3 kinase gamma inhibitor. And this has a very interesting effect on cardiac function, because you see here we measured again contractility in these hearts where adrenergic receptor density is significantly increased. We also see that contractility improves. And of course, in our mice, where PI3 kinase gamma is catalytically inactive, we have that adrenergic receptors are still normal, don't get downregulated after some time, and actually contractility stays. And as I showed you before, these mice do not develop heart failure. So the point here is that in heart failure, PI3 kinase gamma activity is needed to produce PIB3 that in turn causes shedding of adrenergic receptors. But this also means that in healthy conditions, PI3 kinase gamma activity has to be selectively regulated. And as we see this PI3 kinase gamma activity going up in heart failure, this means that probably in healthy conditions, PI3 kinase gamma activity has to be turned down. And this is actually the case, because we treated cardiomyocytes, hearts, in explanted hearts, in vivo hearts, with different ways that cause adrenergic receptor activation. And in all situations, you see that when this is acutely achieved in a healthy condition, PI3 kinase gamma catalytic activity is significantly reduced. So basically, we think that in healthy hearts, PI3 kinase gamma activity has to be controlled tightly, and basically, there's something that causes inactivation of this signaling enzyme. So as I told you, we stimulated this inactivation with adrenergic signaling, and so we started to think about potential mechanisms that could control this enzymatic downregulation. And so you all know that beta adrenergic receptors activate the cyclic AMP pathway, adenylyl cyclase, and the cyclic AMP is known to activate PKA. So we started to think maybe we have an interconnection between PI3 kinase signaling and cyclic AMP signaling. So we started to think of, or let's say we started to hypothesize that PKA could be involved in the inactivation of the catalytic activity of PI3 kinase gamma. And that is why we ran a series of simple experiments. We put different PKA inhibitors in our assays. We used the PKI, we used a small molecule H89. And in all cases, you see that we actually can restore PI3 kinase gamma activity to the level that is achieved before stimulation. So basically, we sorted out, we figured out that PKA is somehow involved in the control of PI3 kinase gamma activity in a sort of a negative feedback loop. So you see, we also showed that PKA can directly phosphorylate the PI3 kinase gamma. And we also showed that phosphorylation of PI3 kinase gamma by PKA is able to downregulate the enzymatic activity. We mapped the phosphorylation site. It's this threonine 1024. As you see here, this is the catalytic loop. So basically, this threonine 1024 is a ridge just outside the catalytic pocket. So basically, we think that there are good structural bases for this phosphorylation to inactivate PI3 kinase gamma activity. So in basal conditions, PI3 kinase gamma is turned down, and it's turned down by PKA. PI3 kinase signaling, it's not turned down. So this was a bit of a nightmare because we had a signaling that is switched off, but still the literature was reporting that the classical PI3 kinase pathway can be activated by adrenergic signaling. And actually, having different mice that we produced that express mutant isoforms of PI3 kinase gamma, we were able to dissect the specific PI3 kinase isoform that is involved in activation of AKT, so the classical PI3 kinase pathway. And we think, of course, this is not PI3 kinase gamma. It's another isoform. It's PI3 kinase beta. But still, we were intrigued by the interaction between PI3 kinase gamma and PKA. Usually, PKA is very much compartmentalized in cardiomyocytes. So the spatial localization of PKA is specifically controlled by a series of scaffold proteins that are called ACAPs, or A kinase anchoring proteins. So we were thinking that perhaps there must have been such a protein controlling the crosstalk between PKA and PI3 kinase gamma. But some data we produced some time ago were also suggesting that probably the mechanism was a little bit different. So some years ago, we reported that mice that lack PI3 kinase gamma and mice that express a catalytically inactive PI3 kinase gamma have extremely different phenotypes in terms of responses to aortic constriction. So while knockout mice without the protein are in a really bad shape, and you see here all these white spots, these are areas of cardiomyocyte loss, like a lot of infarction sites. But mice that express the protein, although without any catalytic activity, the hearts are absolutely normal. So this was telling us by a genetic means that PI3 kinase gamma was not only a kinase, but also something else, and probably something related to a scaffold activity. So we started again to go back to our PKA studies, and we actually were able to show that PKA and PI3 kinase gamma can directly interact. We measured this interaction by different means. Probably the best or the most convincing experiment comes from the biocore analysis with purified proteins. We were able to measure a KD. The KD is not really fantastic. It's in the low micromolar range, but this is known to be sufficient for a lot of these scaffold proteins that actually bind PKA and localize PKA-mediated signal transduction. So this was telling us that somehow PI3 kinase gamma is an ACAP and is able to bind PKA. But usually A caps are part of larger complexes. So you see here that the A caps are part of complexes containing different signal transduction enzymes. So we have already now a complex containing 2 different kinases. We started to think that maybe there was more to it, and there was something perhaps more sufficient to explain these big differences in phenotypes comparing the knockout and the kinase-dead. So we were able to immunoprecipitate the PI3 kinase gamma with pKa, but also with some phosphodiesterases. So the story gets a little bit more complicated. So now we have 2 kinases and even a phosphodiesterase. Phosphodiesterases are known to block or destroy cyclic MP. So we have pKa that blocks PI3 kinase activity, but we also have phosphodiesterases that block pKa. So basically, this is the model we think of. So PI3 kinase is an enzyme, but it's also a scaffold protein. It interacts with phosphodiesterases just to bring or to localize pKa next to them so that pKa can actually activate phosphodiesterase activity, cyclic MP destruction, and so a negative feedback loop on itself. And that is why we have these effects on contractility. And actually, when we measure the cyclic MP contents in our cardiomyocytes with a FRET sensor, you see here that in the absence of PI3 kinase gamma, cyclic MP gets significantly elevated, while the blue and the green lines, the blue is a wild-type control, the green one is cardiomyocyte expressing a catalytically inactive PI3 kinase gamma. There you see that cyclic MP levels are staying pretty normal. The interesting thing is that in the knockout, where the scaffold activity is lost, cyclic MP increases, but also the kinetics is shifted to the right, indicating that it's the destruction of cyclic MP that is affected, and indicating that somehow it is the effect of the scaffolding effect of PI3 kinase gamma to pKa that allows the activation of phosphodiesterase in the cyclic MP destruction. And we think that accumulation of this cyclic MP in our cardiomyocytes is the reason for this tremendously strong phenotype we detected in the knockout. So basically, the model is the following. We have a PI3 kinase gamma that is a scaffold protein. I told you about it, but it's also a kinase. We have a crosstalk between 2 pathways, the cyclic MP pathway and the PIP3 pathway. This pathway, the PIP3 pathway, is somehow connected to adrenergic receptor internalization, and we think that if we can block the catalytic activity of PI3 kinase gamma in a selective way, then of course we might protect the hearts from maladaptive remodeling and failure. And this is part of the story I told you just came up in molecular cell, and we got the cover as you see there. So I would like just to end up here mentioning the people in the lab who contributed to this work. The PI3 kinase as an ACAP was the work of a very talented PhD student, Alessia Perino. The chimeras were done by Irene Franco and Federico da Milano. Federico already left the lab for Harvard, so it's a real disaster. But anyway, we are trying to, of course, recover from this. And I would like to also to thank all these collaborators around the world. But I will also would like to thank a really special collaborator, so Leonardo da Vinci, that helped us to get the cover in molecular cell. Thank you very much.